Welcome to our nocturnal laboratory of gentle discoveries. Here at Dreamtime Science, we transform complex concepts into lullabies for the curious mind. You know, I've been thinking about how tiny, invisible tools inside our cells can actually be guided to make precise changes, almost like nature's own little editor. There's something remarkable about CRISPR, this natural mechanism that scientists have learned to use to gently rewrite the story of life itself. I can't wait to share how this incredible discovery lets us explore the building blocks of living things in a way that feels both powerful and full of wonder. If this peaceful blend of science and serenity helps ease your evening, consider joining our community of gentle learners. Share your favorite moment of scientific awe in the comments. Take this moment to settle in, wrap yourself in comfort, release the day's weight from your shoulders, and let your breath become as gentle as starlight. In the hush of night, when the world settles into a gentler rhythm, I invite you to imagine a letter, sealed and silent, waiting on the threshold of your dreams. This is not an ordinary letter. No, it is a message from tomorrow, penned in the elegant script of possibility. The paper shimmers faintly, as though woven from moonlight and memory, and if you listen closely, you can almost hear the whisper of genes uncoiling, of secrets waiting to be told. Unfolding the letter, you find that it speaks not in words, but in the language of life itself. Nucleotides, those four humble letters, A, T, C, and G, are the alphabet, and every living thing is a sentence written in their code. For centuries, humanity has gazed in awe at this script, deciphering its grammar, pondering its mysteries, dreaming of what might be possible if only we could learn to write with the same effortless precision. Tonight, let us linger in that delicate space between awe and ambition as we begin to explore a most extraordinary tool, CRISPR. Its name is crisp and unassuming, but it is the quiet thunder at the heart of a revolution. CRISPR, clustered, regularly interspaced, short palindromic repeats, a phrase that rolls from the tongue like a spell, and in a way it is one, a spell to rewrite the instructions of life itself. But before we grasp the quill and begin to edit the letter of life, let us drift through the history hidden within those four letters. Imagine a scientist hunched over a microscope, his breath fogging the glass, his eyes wide with wonder. For decades, genetic engineering was the province of dreams and distant futures, the stuff of science fiction novels and whispered what-ifs. In stories like Gattaca, the future was painted in stark monochrome, a world where destiny was dictated by the invisible hand of DNA, and the spark of human spirit flickered beneath layers of engineered perfection. In Jurassic Park, ancient creatures were conjured from the past, their mighty forms reconstructed from fragments of code, brought roaring into a world that had forgotten them. These stories, thrilling and terrifying in equal measure, danced on the edge of what was possible. They asked us to imagine, what if we could shape life as easily as we shape clay? What if the boundaries between what is and what could be were as thin as a strand of DNA? For years the tools we had were blunt and imprecise, more like axes than scalpels. Early geneticists could move genes from one organism to another, but the process was slow, messy, unpredictable. It was like trying to edit a novel by cutting out whole pages and hoping the story still made sense. The dream of precise, efficient, affordable gene editing seemed just out of reach, a horizon glimpsed through fog. Then, quite by accident, the key was found in the most unassuming of places, the ancient war between bacteria and viruses. In the teeming microcosm beneath our notice, bacteria have been fighting a quiet, relentless battle for survival. Their enemies are bacteriophages, viruses that invade, hijack, and destroy. To defend themselves, bacteria developed a memory of past invaders, a genetic scrapbook tucked away in their DNA. These were the CRISPR sequences, repeating patterns, each interspaced with fragments of viral code. Every time a bacterium survived an attack, it snipped a piece of the invader's DNA and tucked it into its own genome, a warning for future generations. But the true marvel was the system that used these memories for defense. When a virus returned, the bacterium could recognize the intruder's DNA, guided by the sequence it had stored. It summoned the Cas9 protein, a molecular pair of scissors, guided precisely to the matching code. With surgical accuracy, Cas9 would slice the viral DNA, neutralizing the threat. It was a beautifully orchestrated act of genetic defense, evolved over eons, hidden in plain sight. It was only in recent years that scientists realized the potential of this microbial arms race. What if, instead of defending bacteria, we could harness this system to edit any gene we chose? What if we could guide Cas9 to any point in the genome, cut precisely where we wished, and rewrite the code of life? Suddenly, the tools of nature became our own. The script could be edited, revised, even composed anew. CRISPR-Cas9 is, at its heart, breathtakingly simple. It requires only a guide RNA, a short strand of code that matches the target sequence, and the Cas9 enzyme primed to cut. When introduced into a cell, the guide RNA leads Cas9 to its destination, and there, with a snip, the double helix is broken. The cell, sensing the damage, rushes to repair it. In doing so, it offers us a precious opportunity. We can trick the cell into making a specific change, inserting or deleting letters, rewriting the sentence of life. Consider the elegance of this mechanism. In the vast, tangled labyrinth of a genome, stretching over 3 billion letters in humans alone, CRISPR can find a single word, a single letter, and alter it. It is as if one could wander through the Library of Babel, locate a single book among millions, open to a single page, and change a single word, forever altering the story. The implications unfurl before us like a map to lands unknown. Already, scientists have used CRISPR to edit the genes of plants, making crops resistant to disease, tolerant to drought, richer in nutrients. In animals, it has been used to cure blindness in mice, to prevent deadly muscle degeneration in dogs, to breed pigs whose organs might one day save human lives. In the realm of human medicine, the possibilities shimmer with hope and anxiety, correcting the mutations that cause sickle cell anemia, cystic fibrosis, muscular dystrophy. Diseases that once spelled doom now face the prospect of erasure, written out of existence by a few deft strokes. Yet as we stand on the threshold of this new era, the shadows lengthen. For every promise there is a peril, for every step forward a deepening unease. The power to edit life is intoxicating, but it is also humbling. What stories do we wish to write? What risks are we willing to take? In the world of gattaca, genetic selection has become routine, and a hierarchy is built upon the purity of one's DNA. In Jurassic Park, the resurrection of extinct species ends in chaos, a reminder that nature's wisdom is not easily surpassed. These tales are cautionary, but they are also invitations to reflection. The tools we wield are neither good nor evil. They are mirrors reflecting the choices we make. CRISPR is a chisel, and we are the sculptors. The future is not written, but it is being drafted, line by line, in laboratories, clinics, and fields across the world. Let us pause here, in this liminal space between wonder and warning, and consider the profound intimacy of what CRISPR offers. It is not merely the power to cure or alter, but the power to converse with the deepest part of ourselves. Every cell in your body contains a copy of your genome, a story that began billions of years ago and has been passed, generation to generation, through the river of time. With CRISPR, we can reach into this river and redirect its course. We can erase a misstep, amplify a strength, even introduce something new, a gene from another organism, a trait from another world. The prospect is dizzying, almost dreamlike. It is as if the boundaries between science and fiction have dissolved, and we are left to navigate a landscape where anything may be possible. Picture for a moment a child born free from the shadow of inherited disease, her genome carefully edited so that she need never fear the illnesses that haunted her ancestors. Picture a forest of chestnut trees, once nearly wiped from the earth by blight, now thriving again, restored by the insertion of a single protective gene. Picture a world where mosquitoes cannot carry malaria, where crops flourish without pesticides, where the genetic lottery is no longer a sentence but a starting point. Yet, for all its promise, CRISPR is not omnipotent. It is precise, but not infallible. Sometimes the cut is made in the wrong place. Sometimes the cell's repairs introduce errors of their own. The fear of unintended consequences, off-target effects, genetic mosaicism, unforeseen impacts on ecosystems, lingers at the edge of every experiment. And then there is the specter of misuse, the temptation to enhance beauty, intelligence, athletic prowess, to engineer not just health but desire and ambition. The line between therapy and enhancement blurs, and with it our certainties about what it means to be human. The letter from tomorrow is unfinished, the ink still wet. It asks questions we have barely begun to answer. Who decides which genes are worthy of editing? Who ensures the safety, the equity, the wisdom of these choices? How do we balance the yearning to heal with the humility to refrain? CRISPR is not just a tool, but a test, a measure of our vision, our restraint, our compassion. As the night deepens and the possibilities swirl around us like falling leaves, let us listen to the quiet pulse of life. Each heartbeat, each breath is a testament to the resilience and fragility of the code that sustains us. The double helix twists and turns, carrying within it the hopes of a species that has always dreamed of more. Outside, the stars wheel silently across the sky, their light travelling millions of years to reach us. In their glow we glimpse the vastness of what lies ahead, worlds within worlds, stories within stories, all waiting to be written. The letter from tomorrow is an invitation to wonder, to question, to create with care. And as we lay the letter gently aside, unresolved, unsealed, we sense that the true adventure is only beginning. The script of life is no longer immutable, and we, its readers, are now called to become its authors. In the next quiet turning of the night, we will slip deeper into this landscape, exploring the intricate ways CRISPR shapes not only the bodies of living things, but the stories they tell and the futures they weave. For now, let the possibilities drift around you, gentle as the wind, and let the quiet promise of tomorrow linger in your dreams, awaiting the dawn. If you listen quietly, with the patience of 1,000 years, you might almost hear it, the low, intricate hum of the living cell deep beneath the surface of your skin. It is a labyrinth, this place, coiled within every living thing, a maze of world proteins, rivers of cytoplasm, ramparts of lipid membrane. But at its secret center lies a string of letters, a code written in the alphabet of life. It is neither ink nor script nor even a language as we understand it, but a chemical text, a double helix of nucleotides that whispers instructions for the making and unmaking of a living organism. For centuries, this code was unbreakable. It was a cipher so dense, so deeply entwined with the very fabric of the world, that the greatest minds could only glimpse its surface. But now, in this strange, thrilling era, we have found a way to reach into its coils, to read, to alter, to rewrite. Among the tools that have granted us this power is CRISPR, a molecular scalpel sharp enough to cut the very script of life. Yet what seems, from afar, to be a miracle of modern science, simple, swift, almost magical, reveals, upon closer inspection, all the complexity and peril of a labyrinth. To understand the mechanism of CRISPR is to step into the winding corridors of molecular biology, where nothing is quite as straightforward as it seems. Imagine, if you will, a cell, perhaps one of your own, humming gently in the dark. Within its nucleus, the DNA coils and uncoils, a ceaseless ballet of replication and repair. The CRISPR system, though now famous as a tool of human invention, was born not in a laboratory, but in the ancient struggle between bacteria and their viral foes. Long before CRISPR was a buzzword in genetic engineering, it was a humble immune system, evolved by single-celled organisms to defend against marauding viruses called bacteriophages. When a virus invades, it injects its own DNA into the bacterium, an act of molecular piracy that, if unchecked, spells doom for the host. But bacteria are not defenseless. Some have learned to snip out a fragment of the invader's genetic code and tuck it away into their own genome, slotting it into special regions called clustered, regularly interspaced, short palindromic repeats, hence the acronym CRISPR. These stored fragments are more than souvenirs. They are warning flags, molecular mugshots. When the virus strikes again, the bacterium transcribes these records into RNA guides. Each guide is paired with a protein, most famously Cas9, the molecular scissors. Together they patrol the cell, and if they encounter a sequence matching the stored viral DNA, Cas9 is triggered to cut, cleaving the invader's code and halting the infection. It is this system, elegant in its simplicity and devastating in its precision, that scientists have borrowed and adapted, transforming a bacterial defense into a gene-editing tool. But here, in the translation from microbe to laboratory, the myth of simplicity begins to unravel, for the cell is not a blank parchment, and DNA is not a mere string of letters to be rearranged at will. Consider first the act of targeting. To edit a gene with CRISPR-Cas9, one must design a guide RNA that will seek out a precise stretch of nucleotides, perhaps a gene linked to disease or a regulatory region affecting development. The promise is specificity, a guided missile homing in on its target with uncanny accuracy. Yet the genome is vast, a billion-letter epic, rife with sequences that echo one another. Even the best guide can sometimes falter, binding to a similar but unintended site. These off-target effects are the first winding turns of the labyrinth, subtle, often invisible, but carrying consequences that ripple through the cell. When Cas9 finds its target, it cuts both strands of the DNA, creating a double-strand break. This is a wound, a crisis that cannot be left unhealed. The cell rushes to repair it, deploying one of two main pathways: non-homologous end joining, a quick and dirty patch that often leaves scars, or homology-directed repair, a more precise method that can be coaxed to insert new genetic material if a template is provided. In theory, this allows for surgical rewriting. Remove a faulty gene, insert a healthy one, correct a single errant letter. But in practice, the cell's repair machinery is not always so obliging. The repair may be imperfect, introducing small insertions or deletions, or the template may be ignored altogether. The outcome is contingent, probabilistic, a dance between human intention and cellular caprice. The deeper one wanders into this maze, the more one sees the limits of our dominion. Genes do not exist in splendid isolation. They are embedded in networks, their expression regulated by distant enhancers, subtle chemical tags, and the folding architecture of chromatin. To alter one gene is to tug at a thread in a living tapestry, with consequences that ripple outward in unexpected ways. A mutation that seems innocuous in the petri dish may, in the context of a complex organism, unleash a cascade of effects, some beneficial, some harmful, many unpredictable. And so, the myth of simplicity, that gene editing is as easy as snipping and pasting text, collapses under the weight of biological reality. Each intervention is a wager, staked on our incomplete understanding of the genome's interwoven logic. The tools are powerful, the potential vast, but the path is fraught with uncertainty. Yet the allure is undeniable. With CRISPR, we have already cured genetic diseases in cells and animals, engineered crops to resist blight, and even begun to contemplate the editing of human embryos. The very phrase, editing the human germline, carries a tremor of awe and anxiety. For to change the DNA of an embryo is to write not just into one life, but into the lives of all its descendants, a kind of molecular legacy echoing down the generations. Here, in the heart of the labyrinth, we are confronted not just by technical challenges, but by the deepest questions of ethics and responsibility. What does it mean to wield such power? Who decides which traits are worthy of correction, which variations constitute disease, and which are simply the normal scatter of human diversity? As the technology advances, the line between therapy and enhancement begins to blur. Is it ethical to correct a mutation that causes a fatal disorder? Few would argue otherwise. But what of traits that are less clear-cut? Susceptibility to certain diseases, variations in height, intelligence, or temperament. The prospect of designer babies enters the public imagination, stoking fears of a future divided by genetic privilege. Even as we marvel at the possibilities, we must reckon with the limits of our foresight. The genome is not a blueprint, but a dynamic system shaped by evolution and environment. To edit one gene is to intervene in a story billions of years in the making, written in a dialect that we are only beginning to decipher. There are risks we can anticipate, off-target mutations, immune reactions, unintended side effects, and others that lie hidden, waiting to emerge in the fullness of time. The social and ethical landscape is as complex as the molecular one. In some countries, the editing of human embryos is strictly forbidden. In others, research proceeds under careful regulation. The international consensus is fragile, shifting, contested. There are voices calling for a moratorium on germline editing, others arguing for its careful, transparent pursuit. The debate is not merely academic, for already, in secret or in defiance of norms, the boundaries are being tested. Yet even outside the realm of human genetics, the implications are profound. CRISPR has been used to engineer mosquitoes that cannot transmit malaria, to revive extinct genes in vanished species, to create organisms with abilities no creature has ever possessed. Each advance is a step deeper into the labyrinth, where the threads of ecology, evolution, and ethics become ever more tightly woven. And so we find ourselves at a crossroads, poised between wonder and caution. The CRISPR mechanism, for all its apparent cleanness, is not a wand, but a key, one that opens doors into rooms we have never seen, chambers filled with both promise and peril. The scientist in the lab, the ethicist at the forum, the parent facing the shadow of inherited disease, all are wanderers in this maze, each seeking a path through the tangled corridors of possibility. It is tempting to believe in the flush of discovery that we are masters here, that with enough precision, enough knowledge, we might finally chart the twists and turns of the genome, illuminating its every shadow. But the living cell is not a machine to be programmed at will. It is a creature of context, history, and chance. The same edit that brings health in one environment may bring harm in another. The same gene may play different roles in different tissues at different times in life. The more we learn, the more we are humbled by the intricacy of the code and the subtlety of its regulation. Occasionally, the headlines ring with stories of CRISPR's triumphs, of children cured, fields transformed, diseases vanquished. Yet these victories are hard-won, the result of years of labor, of failed experiments, of endless refinement. Behind each success are questions unresolved. What of the changes we do not see? What of the long-term consequences, the secondary effects, the hidden costs? The labyrinth is patient. Its secrets are not easily yielded. And there are deeper questions still, echoing in the silence between base pairs. What does it mean, in the deepest sense, to alter the code of life? Are we caretakers, co-authors, or interlopers? The answers are not written in the genome nor in the laws of science, but in the choices we make as a species, choices that will reverberate long after the first edit is made. The labyrinth of life's code is not a place one exits easily. Each discovery leads to new corridors, each answer to fresh uncertainty. The CRISPR mechanism, for all its elegance, is only a tool, a way to navigate, not to escape, the profound complexity of biology. And as we move forward, step by tentative step, we do so with both hope and humility, knowing that the code we seek to edit is also the code that shapes us. Somewhere, in the quiet heart of a single cell, the double helix coils on, a story still being written, a labyrinth whose end is not yet in sight. The next chamber lies ahead, silent and waiting, its doors still closed. The journey through the code continues, each turn revealing both the power and the mystery at the core of life. Before the age of genes as we now understand them, before the language of adenine, cytosine, guanine and thymine was even whispered in lecture halls, the manipulation of life was already underway, though in a far cruder form. Millennia ago, farmers bred wheat by hand, seeking plumper grains and heartier stalks. Fishermen chose the largest carp to spawn in their ponds. These were the earliest genetic artisans, shaping life not with knowledge, but with observation and patience, nudging the slow river of heredity this way and that, guided by the faint glimmer of desired traits. But the loom was hidden, and the threads were invisible. Centuries passed. The microscope brought the world of the very small into focus, and a Moravian monk named Gregor Mendel, in the hush of his monastery garden, began to count peas, wrinkled and smooth, yellow and green. He tracked their inheritance, and by so doing glimpsed the outlines of rules that governed the passage of traits. Still, the fabric of life was only dimly seen, the machinery unknown. It would not be until the 20th century that the double helix would reveal itself, winding its way across the pages of scientific journals, a structure both elegant and profound in its simplicity. In time, as the structure of DNA became known, so too did the possibility arise for its deliberate alteration. The first tools were blunt, their precision lacking. Radiation and chemicals sprayed shotgun patterns of mutation across the genome, a scatter of chance, as likely to kill as to cure. But as the century advanced, science found its scalpel. Restriction enzymes, discovered in the 1970s, allowed the careful snipping of DNA at specific points. These enzymes, evolved by bacteria to slice up the genetic material of invading viruses, became the first implements in the genetic surgeon's kit. Scientists could, with effort and luck, cut and paste fragments of DNA, splicing genes from jellyfish into mice, from bacteria into corn. The age of genetic engineering had begun, and with it the promise and peril of rewriting life. But these methods, for all their ingenuity, were laborious and imprecise. Genes inserted at random, sometimes in the wrong place, sometimes in many copies. Unintended mutations crept in. The dream of editing genomes with the ease and precision of a word processor remained out of reach. The artisans of genetics, it seemed, still worked with candlelight and rough-hewn tools. It was into this world, ripe with possibility but hungry for precision, that a new story began to unfold, not in the gleaming halls of the world's great universities, but in the humble domain of bacteria. For bacteria, ever beset by viruses, those ancient and tireless predators, had evolved a strange and subtle defense. Within their genomes, researchers found odd palindromic sequences, clusters of repeating DNA punctuated by unique spaces. These were christened clustered regularly interspaced short palindromic repeats, or CRISPR for short. At first, CRISPR sequences were curiosities, genetic oddities noted in passing in bacterial genomes. But as scientists looked closer, they noticed something peculiar. The unique spacer sequences nestled between the repeats bore a striking resemblance to fragments of viral DNA. It was as if the bacteria were keeping records, genetic mugshots of viruses that had attacked them in the past. The spacers were a memory, a molecular scrapbook of battles survived. The function of CRISPR remained opaque until a series of clever experiments conducted at laboratories far-flung across the world began to illuminate its role. In the mid-2000s, researchers such as Francisco Mojica in Spain and later Philippe Horvath and Rodolphe Barangou working in the dairy industry pieced together the mystery. They showed that when bacteria survived a viral attack, they often acquired a new spacer matching the invader's DNA. If that bacterium encountered the same virus again, the CRISPR system would recognize and destroy it, wielding a molecular scissor known as Cas, a CRISPR-associated protein. This was bacterial adaptive immunity, a memory of invaders written into the genome, wielded as a weapon to defend against future assaults. Cas proteins, guided by RNA transcripts of the spacer sequences, would patrol the cell's interior. When a matching viral sequence appeared, the Cas protein would slice the DNA, neutralizing the threat. What had begun as an esoteric curiosity was now seen as a sophisticated immune system, one that could be harnessed and perhaps repurposed. Even as these discoveries unfolded, the possibility of co-opting this bacterial machinery for deliberate genetic editing shimmered at the edge of the scientific imagination. The breakthrough would require not just insight, but the patient, methodical work of many hands and minds. It was Emmanuel Charpentier, working in Umeå, Sweden, who first isolated the key components of one such system, the Cas9 protein and the dual RNA structure that guided it. In a 2012 collaboration with Jennifer Doudna at the University of California, Berkeley, the two teams embarked on a series of experiments that would transform the field. In their hands, the CRISPR-Cas9 system was reconstituted in a test tube, the components purified and assembled like the pieces of a clock. They found that they could program the system to target virtually any DNA sequence simply by changing the guide RNA. Where previous genetic tools were cumbersome and limited, CRISPR-Cas9 offered speed, efficiency, and above all, precision. It was as if the artisans of genetics had at last been handed a brush fine enough to paint on the smallest of canvases. The elegance of the CRISPR-Cas9 mechanism lies in its simplicity. The Cas9 protein forms a complex with a guide RNA, which is designed to match a particular DNA sequence. Like a bloodhound scenting a trail, the complex roams the genome, seeking out its target. When the matching sequence is found, Cas9 cleaves the DNA, introducing a double-strand break. The cell, confronted with this rupture, attempts to repair the damage, sometimes introducing small errors, sometimes allowing a new stretch of DNA to be inserted. In this way, genes can be deleted, altered, or even replaced with exquisite accuracy. The path to this discovery was neither straight nor smooth. It was marked by false starts, rivalries, and the slow accumulation of knowledge. The story unfolds in crowded laboratories where graduate students and postdocs pipetted clear liquids into tiny tubes, peered anxiously at the faint bands on gel electrophoresis sheets, and debated the meaning of ambiguous results. Each experiment was a puzzle, each failure a clue. The artisan's touch was everywhere, in the careful design of RNA sequences, the tuning of reaction conditions, the patient repetition of protocols late into the night. Elsewhere, in the world of mouse embryology, Feng Zhang and his colleagues at the Broad Institute raced to adapt the CRISPR-Cas9 system for use in animal cells. Their success, announced within months of the Doudna-Charpentier publication, demonstrated the system's versatility. The same bacterial immune mechanism could be wielded in the cells of plants, animals, and even humans. The race was on, and laboratories worldwide scrambled to adopt the new tool, each seeking to unlock the secrets of their chosen organism. As CRISPR spread, new variations and enhancements appeared. Scientists in Japan, China, and Europe tinkered with the Cas9 protein, altering its structure to improve specificity or to expand its range. Others discovered alternative Cas proteins like Cas12 and Cas13, each with their own unique properties, each opening new doors to the manipulation of genetic material. The CRISPR repertoire grew steadily, the artisan's palette now rich with colors and shades once unimaginable. Yet amid this rapid progress, the spirit of curiosity and collaboration endured. The making of genetic artisans was not only a matter of technical skill or cleverness, but of a certain humility before the complexity of living systems. For every triumph, there were setbacks, off-target effects, unexpected cellular responses, ethical dilemmas posed by the very power of the tools now in hand. The artisans learned to move carefully, to weigh each possibility, to listen to the silent language of cells. In university basements and biotech startups, CRISPR became the linchpin of 1,000 projects. Crops resistant to blight, mosquitoes unable to carry malaria, pigs with organs suitable for transplantation, the correction of single-point mutations in the genes of children with rare diseases. The possibilities multiplied, each more dazzling than the last. And yet, each required the same underlying skills: the design of precise guide RNAs, the crafting of delivery systems to smuggle the CRISPR machinery into the nucleus, the monitoring of cellular responses, all under the watchful eye of the artisan scientist. Even as the field advanced, the mechanics of CRISPR continued to reveal new subtleties. Researchers found that the position of the guide RNA mattered, that the local chromatin landscape, the way DNA was packaged and marked with chemical tags, could influence the efficiency of editing. Some cells resisted the changes, others embraced them too readily. The act of editing was less like typing on a keyboard, and more like coaxing a living tapestry to accept a new thread, woven with patience and care. Meanwhile, the story of CRISPR's discovery became a parable of scientific culture, a tale of open data and fierce competition, of patent disputes and Nobel Prizes. The artisans of the genome were not immune to the temptations of recognition and reward, but for many, the true satisfaction lay in the work itself, in the thrill of seeing a band appear at just the right place on a gel, in the moment when a colony of bacteria glowed green under ultraviolet light, proof that a gene from a jellyfish had found a new home. The refinement of genome editing tools did not end with CRISPR-Cas9. New generations emerged, each layer building upon the last. Base editors arrived, capable of changing a single DNA letter without breaking the strand. Prime editors followed, able to write new information with even greater precision. The artisans became architects, able to remodel the blueprints of life with increasing subtlety. And yet, for all the sophistication of these tools, the heart of genetic editing remained the same: a dialogue between the scientist and the living cell, a negotiation with the deep logic of biology. The artisans worked not only with molecules, but with time, with the rhythms of cell cycles, the intricacies of development, the slow unfolding of phenotype from genotype. Their craft was both ancient and new, rooted in the patient selection of traits by the earliest farmers, yet transformed by the ability to reach into the very machinery of inheritance itself. Late at night, when the hum of freezers and incubators filled the lab and the city outside lay still, the genetic artisans continued their work. They checked cultures, adjusted protocols, recorded observations in notebooks worn soft by use. Each step, each result, was another brushstroke in the vast mural of biological understanding. Their hands, steady and sure, moved with the confidence born of practice, but their eyes still shone with wonder. There were moments of frustration, of course, experiments that failed for reasons unknown, cells that refused to grow, DNA that degraded in the freezer. But there were also moments of awe, The sudden realization that a gene had been precisely altered, that a disease-causing mutation had been erased, that a living organism had been reshaped in ways previously confined to the realm of dreams. The making of genetic artisans was, and remains, a story of perseverance and imagination. It is a story written not only in the annals of science, but in the quiet triumphs that play out each day at the lab bench. For every headline and scientific award, there are countless unheralded acts of creativity and care, the careful calibration of a pipette, the patient troubleshooting of a protocol, the shared excitement of discovery among colleagues. And so the work continues, the story unfolding, the artisans at their benches and computers, their minds turning over new possibilities, their hands preparing the next experiment. The tools grow sharper, the techniques more refined, but the essential challenge endures: to shape life with precision and respect, to understand the deep language of genes, to walk the fine line between invention and humility. Beyond the walls of the laboratory, questions gather like clouds on the horizon. What will we choose to change, and what will we choose to preserve? As the artisans refine their craft, as the tools of genome editing spread ever outward, the next chapter will ask not only what is possible, but what is wise. In the hush of the lab, as another experiment is set in motion, the future of genetic artisanship hangs suspended, awaiting its next artist's touch. A hush falls over the laboratory, the quiet hum of machines and the gentle tap of fingers on glassware fading into a deeper silence. Here, in this sanctuary of science, a question lingers, weighty and elusive. What does it mean to possess the ability to rewrite the code of life? In the soft glow of lamplight, surrounded by the intricate diagrams of genes and the luminous screens displaying the twists and turns of DNA, we find ourselves standing at a threshold, gazing into an uncertain future. The age of CRISPR is not merely an era of technical prowess. It is, in many ways, a return to a primeval garden, an Eden of possibility and peril, where humanity is both gardener and seed. There is a peculiar stillness in this new Eden, a suspenseful sense that something profound is about to unfurl. The double helix, once a symbol of the unchangeable fate inscribed in our cells, is now a scroll upon which we etch our intentions. With each precise cut, each careful repair, we are not only altering the fabric of life, but also weaving ourselves more deeply into the story of creation. The line between discovery and invention blurs, and with it the boundary between what is given and what is chosen. The mythic fruit of knowledge hangs heavy on the branch, and the taste of its promise is bittersweet. It is tempting, in the glow of scientific triumph, to see the genome as a puzzle to be solved, a machine to be optimized, yet step back into the hush, and the mystery of life persists. Each cell is still a world unto itself, each gene a word in a language we are only beginning to speak. The more we learn, the more the mystery deepens. How from a single fertilized egg an entire human being unfolds, bones and sinew, memory and longing, laughter and sorrow. The code is not merely a sequence of letters, it is a symphony played out in the living theatre of time. The power to edit this code is, at once, humbling and terrifying. With CRISPR, the inheritance of generations can be rewritten in a single moment. Disease may be erased, yes, mutations smoothed, suffering softened, but the same hand that heals can also harm. It is a tool that does not distinguish between wisdom and folly, compassion and ambition. The genome is not a canvas for idle doodling. It is the inheritance of the species, and every alteration ripples outward, unpredictable as the weather, persistent as memory. Pause and consider the paradox. We are the product of evolution, shaped by millions of years of blind chance and necessity. Yet now we stand poised to direct our own evolution, to become both author and character in the grand narrative of life. the ethical questions rise and swirl, unsettling as a flock of startled birds. Should we correct the mutations that cause suffering, or is there wisdom in accepting the limits of our biology? If we can enhance intelligence, strength, or longevity, are we obligated to do so, or do we risk losing something precious in the quest for perfection? What of diversity, of the wild, unpredictable variety that is the wellspring of creativity and resilience? In a world where genomes can be tailored and traits selected, will we cultivate a garden of infinite richness, or a monoculture stripped of surprise? To contemplate these questions is to recognize that CRISPR is not merely a technical revolution, but a philosophical one. It requires us to revisit ancient debates, the nature of free will, the meaning of justice, the structure of the good life. The power to edit life is, in a sense, the power to define what it means to be human. Do we see ourselves as masters of our destiny, or as caretakers of a fragile inheritance? Is the genome a birthright or a responsibility? And as we reach for the tools of creation, who do we become? The garden of possibility is lush but shadowed. Imagine for a moment a world where genetic disease is a memory, where children are born free of the burdens that haunted their ancestors. The suffering of cystic fibrosis, the relentless decline of Huntington's, the cruel unpredictability of sickle cell anemia, each potentially consigned to history. The relief is immense, the gratitude boundless. Families gather in bright rooms, children run without pain, and the spectre of fate recedes. Here, CRISPR is an instrument of mercy, a fulfillment of the ancient hope that science might one day heal what nature cannot. Yet, as the shadows lengthen, other possibilities emerge. What of enhancement? The temptation to not only cure, but to perfect, is ancient and persistent. If we can rid ourselves of weakness, why not also strive for strength? If intelligence can be sharpened, why not reach for genius? Here, the path grows tangled. The boundaries between therapy and enhancement, between healing and improving, are not drawn in the genome. They are drawn in our hearts and minds. The risk is not only technical, but moral, that in our quest for betterment, we lose sight of the dignity of imperfection, the humanity of limitation. History offers cautionary tales. The eugenics movements of the 20th century, draped in the rhetoric of progress, became engines of exclusion and cruelty. The desire to improve the species, untethered from humility and justice, led to horrors that still haunt our collective memory. CRISPR is not eugenics, but the questions echo nonetheless. Who decides which traits are desirable? Whose vision of perfection prevails? In a world of editable genomes, will we celebrate difference, or will we narrow the spectrum of what is acceptable, what is valued, what is loved? The garden's beauty is in its diversity, in the riot of forms and colours that defy prediction. So too with humanity. Our differences, genetic, cultural, personal, are the source of our strength. The challenge of CRISPR is to wield its power without diminishing that richness, to cultivate a future where the full range of human possibility is cherished rather than pruned. Responsibility, then, is the true heart of the matter. To edit the genome is not simply to wield a tool, it is to accept stewardship over the future. The choices made in laboratories today will echo through generations yet unborn. The weight of that obligation is immense. It requires humility, a recognition that our knowledge is partial, our foresight imperfect. It demands vigilance to guard against the easy seductions of power and pride. And it calls for justice to ensure that the benefits of genetic technology are shared widely, not hoarded by the fortunate few. Consider the spectre of inequality. If the fruits of genome editing are reserved for those with wealth or privilege, the gap between rich and poor may widen into a chasm. The ability to pay for health, intelligence, or beauty encoded at the level of DNA could ossify social divisions, transforming accidents of birth into permanent barriers. The spectre of a genetic underclass is not science fiction. It is a warning, a reminder that technology alone cannot solve the dilemmas of justice and equity. The garden must be tended with care, its gates open to all, lest it become a wilderness of exclusion. There are other responsibilities, quieter but no less profound. The relationship between parent and child may be transformed as genetic choices become part of the calculus of love and hope. The act of bringing a new life into the world is already a leap of faith, a gesture of trust in the future. With CRISPR, that leap is accompanied by new decisions: what to alter, what to leave untouched, what to accept as the gift of chance. The bond between generations, once shaped by the randomness of inheritance, now bears the imprint of deliberate choice. The meanings of kinship, identity, and legacy shift and shimmer in this new light. And what of the non-human world? The reach of CRISPR extends beyond the human genome, promising to reshape the very web of life. Mosquitoes engineered to resist malaria, crops fortified against blight and drought, species revived from the brink of extinction, each is a testament to the power of genetic editing. Yet each also raises questions about our relationship with nature. Are we caretakers or conquerors? Do we have the wisdom to intervene in ecosystems whose complexity exceeds our understanding? The garden is not ours alone. Its balance is delicate, its lessons hard-won. To navigate this new Eden is to walk a path between awe and caution. The promise of CRISPR is immense, but so too is its unpredictability. The genome is not a static blueprint. It is a living, evolving system shaped by forces we only dimly perceive. Our edits may have consequences that ripple far beyond our intentions, echoing through populations and across generations. The humility to recognize the limits of our knowledge, the patience to proceed with care, and the courage to face uncertainty, these are the virtues demanded by the age of gene editing. Amidst the questions, one truth endures. The mystery of life is not diminished by our understanding. Even as we probe the depths of the genome, as we decipher the grammar of genes and sculpt the contours of heredity, the wonder remains. The dance of molecules, the emergence of consciousness, the capacity for love and grief, these are not explained away by the code, but rather illuminated by it. To edit life is not to end the mystery, but to enter more deeply into its unfolding. In the quiet of the laboratory, as night deepens and the stars wheel overhead, the garden of possibility stretches on. The tools of gene editing lie ready, precise and powerful, awaiting the wisdom of those who would wield them. The questions posed by CRISPR are as old as humanity itself. Who are we? Who do we wish to become? And what will we choose when the boundary between what is and what could be is no longer fixed, but fluid as water, open as the sky? The first chapter of this new story is only just being written. The genome, once a sealed book, now lies open before us, its pages blank, its future unwritten. The responsibility is ours, to write with care, with reverence, with an abiding sense of wonder. The mystery beckons unresolved as the garden of possibility awaits the careful tread of those who would nurture its promise, and the world turns quietly beneath the stars, breathing in the darkness, waiting for dawn. As our journey through the delicate dance of CRISPR gently drifts to a close, may the marvel of editing life shimmer softly in your thoughts like a comet tail fading into the hush of space. Rest easy beneath the cosmic canopy, and when you're ready to wander among more Dreamtime mysteries, we'll be here, waiting to drift with you once again.